Today we're going to be doing an engineering experiment where we're going to engineer bacteria to create a smell. This is the Smell It kit. And if you look in the Zero to Hero guidebook, go to chapter 6, you're going to see a little bit more information about what's going to go on in this experiment, all right? So in this Going Deeper 6.1, it shows you a bit about the chemical reaction, describes a lot more in depth here. So before you get going on this experiment, please do check out the Zero to Hero book so you know what's going on at the chemistry level. And also, it's important to note that the Smell It kit contains 2 different baggies, which is a bit different than the more entry-level kits. Bag 1 in general is the genetic engineering experiment, where you're engineering the bacteria with a plasmid to do the function you want. In this case, it's to create an enzyme that's going to help us make the banana smell. If you've done prior engineered kits, the plasmids in those cases are to generate a cool color, right? A color pigment. And for today's video, we're only going to be going through bag 2. So if you haven't done an engineered kit, please go to our YouTube video, search for engineered kit, and go through that entire video so that you know exactly how to do bag 1, right? And it's very similar. Again, the only difference is really the plasmid that you're using. But I do urge you to do an engineered kit first because it's going to allow you to practice and increase your chances of success in the Smell It kit. Okay? So for the rest of the video, we're going to be talking about doing bag 2, which assumes that we've already got our engineered cells from bag 1 experience, and we're going to set up the smell changing reaction. All right. And another important part of this bag 2 is actually a freezer bag. So with your kit, you should have also got a little baggie that should have been put in the freezer. And you're also going to need this for the bag 2 part we're going to cover. So let's get ready to go with creating some banana smell. All right, so let's open this up here. And we're going to separate bag 2. That's what we're going to be using. I'll keep this here. I'll probably refer back to it a little bit. So I'll keep it nearby. We are going to need your freezer bag in a little while. We don't need it immediately, but pretty soon, so we're going to have that handy. It's okay to keep it at room temperature prior to your experiment. You'll want to keep it in the freezer for longer term storage though, okay? Now, we're going to need a pen right away. So let's open up this bag here. Let's see what we got. We've got some Petri dishes, which we've used before. We've got a stencil. Actually, you'll notice that this stencil is a little bit different than in the engineered kits or in bag 1. Just pull that over here. All right. One's yellow, one's reddish orange, and then also the pattern in which you're streaking is a little bit different. Okay. In the engineered style of stencil, the goal is to try and get individual colonies, and so you use the triple streak method, right? Then you use those individual colonies for doing your engineering experiment, right? In this case, for this advanced kit, our goal is to really spread as much bacteria across the plate as we can, and so we call this a double streak, where you're not getting a new loop like you do in the engineered kit, but rather you're dipping in the cells, your engineered cells, and you're streaking it across the plate. You might touch it on the cells again, and then do another double streak across, or just streak both ways. And then at the end, this will create a lot of cells that are all the way across the plate. And then they'll grow, and they'll be able to do the chemical reaction that we're looking for, okay? We've got a day 1 baggie for this, which is going to be used to do some streaking of cells and making the plates. Classic sterile water. And then we got a bunch of loops here. We've got both blue loops and we've got yellow loops. Okay. Classic. Okay, so one thing you'll notice that's also different within the bag 2 of the Smell It kit is that there are Petri dishes with these little white papers in there. And these papers are pretty important because they're going to help make the substrate, or the chemical that we're wanting our cells to convert into the smell. These little pieces of paper are going to kind of keep that substrate at the bottom of the Petri dish so that it slow releases into the agar. Okay, so that's really important. And you also notice that only 3 of the 4 Petri dishes have that. Okay, and also notice within your freezer bag, the substrate, there's only 3 tubes. So you can probably guess that 1 tube for each of these little pieces of paper plates. Okay, and then the other one is used as a different kind of control. Pull those out, get them ready, and the next step is we need to label. Alright, we're set up to do some plate labeling, and this is really important because it'll help us plan the experiment in advance. This is a little bit more complex than an engineer kit that you might have done in the past because there are several controls, all right? In the engineer kits, there's a experimental sample, right? There's a positive control sample, right? And a negative control sample that you're running. In this case, you do that with bag 1 of the Smell It kit, right? So you do something similar, but in this case, with bag 2, we have a couple more controls. So it's really important that we label the plates and understand what's going to go in every plate before we get going in this experiment. So I'm looking here in the book, and you can also find this in the instruction manual. One thing to note is that it's kind of difficult to see here, but there's some little faint blue circles which indicate these little pieces of paper that are used to, again, slow release the substrate. Okay, and just for some notation here, IA here stands for isoamyl alcohol. Okay, you'll notice that the 3 plates with the 3 little very faint blue pads here, for these 3 here, they all have IA in them. And as a reminder, IA stands for isoamyl alcohol, which is the substrate that we're going to add to the plates so that they'll diffuse into the agar, ultimately up to the surface where you've streaked bacteria. And if the bacteria has the right enzyme, which is ATF1, this pink abbreviation here, then it'll convert isoamyl alcohol into the banana-smelling product. Okay, so you'll see 3 tubes of isoamyl, and there's 3 plates that have these little papers in them. Okay, so yeah, let's just plan this experiment out. Let's do some plate labeling. One thing to note here is that chloramphenicol, the antibiotic, is in every single one of the plates. Okay? And so there's not unlike the engineer kit where you pour a non-selective and then some selectives. In this case, all of the plates are going to have selection in them. Right? So when you're making your agar, you're going to boil the water, you're going to add the agar, boil again, make sure it's well mixed, and you're going to add the pill at the beginning so that every plate has chlor in it. And this is because you're going to be adding engineered bacteria to every one of the plates. Now, the difference is that some of the bacteria are going to include your engineered bacteria that are now expressing ATF1, again, the enzyme that's going to turn the substrate into the smelly product. Right? You're going to use a negative control E. coli, which doesn't create that enzyme. And the reason you want to do this control is because you want to see whether just regular E. coli is responsible for changing the smell of the substrate into a product, right? In a way, it helps you to say, okay, this one doesn't have the enzyme, and it doesn't smell like bananas, whereas the one with the enzyme smells like banana, okay? There's a third control here, which is a positive control. This includes some cells that you received in the first kit. that are expressing this enzyme, right? So this is just it's a positive control where we know it's expressing ATF1, and this one should smell like banana. Hopefully your engineered bacteria do as well, but this is a positive control. And then lastly, we've got another negative control where we've got E. coli with the ATF1. So again, this is the engineered E. coli to produce the enzyme, and you can use your E. coli that you've engineered, or you can use the positive control E. coli that's engineered, and you put it on a plate that doesn't have the substrate. Okay? And what this negative control will tell you is, does the bacteria itself with the ATF1 smell like bananas? Because now there's no substrate for the bacteria to turn into the banana smell, right? So this control is just seeing whether the bacteria themselves smell like bananas. So yeah, that's the rationale behind the different controls. And so we're going to label these plates now. I'm going to have, I guess we can put E. coli with, I'm going to put a little W slash there, ATF1. So that's one component that's in there. And then we're also going to add isoamyl to this, the substrate, right? And then let's also just put a C for chloramphenicol. Even though we know they're all going to have chlor, we're going to put a C here because, again, you want to be able to communicate this to your peers. You want them to know if there's a C on a plate, that means it's got chloramphenicol in it. All right, so that's plate one. Next one here is just regular, it's E. coli. There we go, get the pen working. It's E. coli. It has a plasmid in it so that it can grow on selective media, but it doesn't have the ATF1 in it, right? So then, therefore, it shouldn't convert the isoamyl into the smell. Right? So this is, we'll just put E. coli here, and we're going to go plus, and then this one is going to have isoamyl plus, and we're just going to add a C there, okay? So this is a negative control. We'll even write, to be very clear, negative control. And actually at the first one here, let's put the E in brackets, or we can even put in brackets, this is our experimental. And so this is the E. coli that you've engineered. Okay, it's your experimental sample. That's what's going to go on here. The second plate here, you're going to use the negative control cells that are provided. They're actually provided within this baggie that comes with the day 2 bag. Let's move on to the third one here. So we're going to have E. coli. This is also going to have with ATF1, and just put that in brackets. Plus, we're also going to add the isoamyl substrate to that one. Okay, and we can also say chlor. It's also going to have chlor in it. And then we're going to put another bracket here that just says plus control. Okay, and the difference between this one, because these both have the E. coli with ATF1, right? The only difference is that this is your experimental sample, meaning you've engineered these cells. And this one is the positive control cells that you received in the bag 1 kit, okay? So those are the ones that should have already been engineered with ATF1 properly, and they're just there to confirm, hence positive control. Okay, and then we got the fourth plate here, where we're adding E. coli with ATF1. Let's put that in brackets again. And this one doesn't have any isoamyl in this one. Plus just the chlor that goes in it. and we're going to put in brackets minus control. Okay. And so just a reminder, the coli with ATF1, these could be your engineered cells, or you can even use the positive control cells, whichever one you choose. And again, this doesn't have isoamyl in it because this negative control is to test whether the bacteria themselves smell like banana, okay? Which hopefully they don't. Good, so now we've labeled our plates. You can see this is a far more complex experiment than the engineer kit, but hopefully you got through that, and if there's any confusion, there's more information in the book that I urge you to read through and make sure you understand prior to the experiment, okay? And even have a discussion with some of your classmates or your teacher just to make sure you're wrapping your mind around why we're using each one of these controls, and what is the rationale and expected outcomes if the experiment is working nominally. We are going to talk about get prepared for pouring these plates because, again, unlike the engineer kit, which is a pretty straightforward protocol, this one's a little more tricky because we need to add substrates underneath these little papers. You can already see they want to stick to the top of the Petri dishes a little bit due to static electricity. Okay? So one way to get around this is if you've got a little bit of water, you can even put your hands in the sink, right? Get them a little bit wet, and you can actually just kind of put a little bit of wetness on the Petri dishes, and that wetness will get rid of most of the static electricity. I'll do that in a moment, but I'm going to first just talk about what we're going to do, because once you get your agar ready, things kind of happen a little bit quickly in this experiment. All right, so you can see we've got not so much, it's about 40 microliters of liquid, so that's a pretty small volume, right? And we're going to be using this little pipette here. We're going to draw up the liquid in the pipette. We're going to try and get all of it into the pipette, and we're going to pipette it onto the Petri dish, and then we're going to move over either with a loop or with the end of this pipette tip. Just move the little paper over top of the isoamyl and kind of like press it down just so that it's absorbed in a little bit and it's underneath, okay? And again, the reason we're doing this is because the isoamyl, we want it to slow release and dissolve into the agar so that we get a sustained smell. We don't want it to just evaporate all right away, because after incubating these for a couple of days, then there will just be no substrate left, right? So this helps to slow release that substrate so the bacteria that are going to grow on the surface have a chance to absorb it into the cells, and then the enzymes can be active and catalyze that reaction. Okay, so to talk this through again, we're going to kind of move these papers onto the side. I'm going to try and get this one off for the actual experiment. There we go, that's a little bit better, so it's off to the side. This one too, I'm going to destatic these with, again, a little bit of water. Move to the side. I'm going to, that's actually probably where we'll get that all prepared. And then we're going to make our agar, and doing this is very similar to how you've done it in the past. So we're going to boil the water, the microwave, rolling boil. You're going to pour in your agar, give it a good swirl, make sure it's mixed in. You're going to boil it a little bit more. You want it to give a nice foam. Make sure it's nice and hot so that the agar part of this mixture dissolves. Give it a good stir or swirl. And then we're going to add the antibiotic, the selection, the chloramphenicol. And add the pill, just like the engineer kit. And we're going to swirl it again until this dissolves. All right, and then we're going to just let that sit to the side. We can't wait too long because we don't want it to solidify inside the bottle. So once that step is done, we're then going to move on to using the little pipette. We're going to pull in the substrate, You might want to not necessarily squeeze all the way, because then if you try and pull it up, it's all going to like shoot up into the little bulb area here. So you might just want to squeeze a little bit. Okay, not a full squeeze, just squeeze a little bit. And then you'll draw that in to this little pipette. You'll set the tube aside. You'll put it on the plate, and again you'll move the little paper over top and give it a little press to allow the isoamyl to soak in a little bit. And then you'll do the same thing with the other two tubes in the other plates. And the one without the paper, it doesn't get any substrate, right? Because again, we talked about that control. Right. And once the isoamyl is underneath, we'll then go ahead and pour the agar on top. And you can either use a loop to help hold down these little papers, or you can use a little pipette, because the general idea is as you're pouring, this little paper is going to want to float a little bit. And so what you can do is hold it down a little bit. Of course, it's going to be in the middle over top of the iso like this. You can hold it down a little bit as you're pouring in. Okay? And just like the engineer kit, you want to fill it up to about the halfway point where this little star is. Great, and you'll do that for each of these plates, and including this last plate here, and you'll let them cool down to room temperature, and then we'll be streaking some cells on top. All right, we're back. The agar has been added. So just like the engineer kit, water boiled, agar added, swirled, boiled again, and you can see it's pretty clear. It's going to be some good agar here. Remember, if you don't boil it enough, it may not gel. So, okay, then we have our chlor antibiotic here. Right, this is what gives it the selection. Just gonna dump that in. All of the plates get it, and then we're going to swirl. Because the substrate is an alcohol, it does evaporate pretty fast. So you've got time. You don't need to super rush here, because it does take the agar 5 minutes, especially if it's in the bottle, like 5 minutes or more to gel. So you've got time, and if you can let it cool down a little bit before it's poured, that's perfectly fine. You can go fast if you want and pour it hot, but it does work a little better if it's a bit cooler. And there we go. You can see the capsule breaking down a bit. Let's see, once in a while maybe. Give it a gentle swirl. You don't want to introduce bubbles, so just swirl it. Okay, so I'm going to give it a little bit of time to dissolve on its own without swirling, and I'm going to get going on these isoamyl substrates, okay? So you might have a tube rack. In fact, I do recommend a tube rack, because then we're pretty sure the isoamyl will all be at the bottom. You can also give a quick centrifuge to these tubes. So you've got a centrifuge, just to make sure all the isoamyl is on the bottom, right? And try and give it the whippet as well. Lean them up a little bit. But again, tube rack is best. Okay, so let's start out here. And pull that over to the side. Actually, it looks like I don't need to get water. It's not too static. Okay, so I squeezed way too much. It wants to go all the way up. Remember, you don't need to squeeze all the way. Here we go. So there it goes. It's on. And let's push that over. So there you go. You can see it was kind of soaked through a little bit. That's perfect. Put the lid back on. And also remember, it does smell. So this is a pretty powerful odor. If you're sensitive to odors, please be careful. And there's also a quick video on wafting on our website, on Amino Labs website, so check that out. You don't want to directly smell concentrated chemical from a container ever. It's very dangerous. Here we go. So you can see it's soaking in. I'll do the third one here. Give this a little swirl. Same thing. You don't need to squish it all the way. Just squish the bulb a little bit. Here it goes. Push that on. There we go. So now we've got it in there. Lid goes on. All right, now we want to try and contain this stuff as much as possible. I'm going to keep using a little pipette here. Let me double check to see. Yeah, it looks to me like the pill is fully dissolved. So I'm going to move on to pouring this in. Let's go ahead one by one. Again, this little paper might want to float, so I'm just going to hold on there a little bit and fill it about half full. Go. Perfect. Next one. I try not to pour directly on the isoamyl because we don't want it to all dissolve away. Go like that. That should be decent. Okay, half full. Third one here. Hold it down a little bit. Half full or so. There we go. Stayed on the bottom, and then the last one, which doesn't have any isoamyl, negative control, half full. There we go. Excellent. So now, I'm just going to put these on because I'm talking. You don't necessarily want to be talking into your Petri dishes, right? So just as a reminder what we have here, we've got a number of plates that we're creating. 3 so far have the isoamyl alcohol substrate under these little paper circles, and those little, because it's underneath and it's soaked in a little bit, when we poured the agar over top, all of the isoamyl doesn't immediately mix in and start to evaporate out. Rather, it's sort of trapped underneath and it's within the paper, and over the course of this whole experiment, several days of growing the bacteria, that isoamyl substrate is going to dissolve out of that little paper into the agar, ultimately up to the surface where the bacteria are, and then ultimately into the bacteria, and then those enzymes that you've engineered them to produce should be able to convert it from isoamyl to the final smelling product, okay? And then the last one here without the isoamyl is a negative control. The bacteria that are engineered with the right enzyme are going to be on the surface, but there's no substrate, right? And so the goal of this negative control, again, is to see whether the bacteria itself produce the banana smell, which hopefully it does not. So we're going to give this a little bit of a wait time, let these cool down, and we'll go get our engineered bacteria from bag 1, positive control bacteria from bag 1, and we have our negative control cells from bag 2. Okay, those should be all the cells we need, and then we already have our loops here, and we can finish streaking the bacteria on the surface using this double streak method this time. All right, see you soon. All right, we're back. Let's just have a quick look here. Might need a few more minutes. Oh yeah, it's only been a couple minutes. They're definitely still quite liquid. While we're waiting here, we'll just quickly check in on the types of cells. So again, we need 3 types of cells for this experiment for all across the controls. We've got our experimental sample. So these are your engineered cells. Hopefully yours looks something like this, or hopefully you just have a single colony. That would be fantastic if you got that. Often in labs, scientists are just hoping to get one colony, and so hopefully you got one colony. And if you didn't, you can share with a colleague. Maybe they have a couple of colonies, or you could use your positive for both of those samples if you like. Up to you. So we need the experimental cells. We need the plus cells. So these were, again, the cells you streaked in the bag 1, the plus cells. Okay. So these are expressing ATF1, the enzyme. Same with these ones, hopefully, right? Should be. And then the third is the negative control cells. So these aren't expressing the enzyme, right? They're just another type of cell that has the plasmid, so they can grow in selective media, but they do not have ATF1, the enzyme, right? And so together, these 3 different cells will help us do all of our controls, and hopefully we'll get some good banana smell. All right, we're back, and let's open up the plate. That's pretty solid now. I think we're good to go. So, because we labeled the bottom of the plates before we did our preparation, this will make streaking the plates a little bit easier and more organized. So maybe we could go ahead and just start with this end plate here. This is going to be an E. coli with ATF1, and it doesn't have any of the substrate in it. And remember, we were able to choose from either the positive controls or our experimental controls. Okay, and ideally, I guess you might use the positive control, just because that's guaranteed to be producing the enzyme. There's really very little reason or no reason why yours shouldn't be producing it. But let's go with that. We're going to use the double streak method here. We're going to put this stencil down. Really, it's just a guideline. You don't need to follow this stencil exactly, but the goal is to try and streak cells all across the plate. There. And I'm going to take the plus cells here. Some of those cells, get them on the loop. And I'm going to streak across. Just turn it about 90 degrees. Not going to get a new loop because, again, I want to get as many cells across the plate as I can. I'm going to streak again, like so. Okay, there we go. So now there should be cells deposited across the plate, and they should grow into a pattern like this, and then as they grow, there's more growth, and these lines get wider, and you get quite a lot of coverage. So that one is done. And I'm going to put this into my bin, which is over here. You should always have your bin with your inactivation bag ready. Okay, let's look at the second plate here. So this is a plate that has E. coli with ATF1, isoamyl, chlor. Got the isoamyl, obviously underneath the paper, and the chlor. We added the pill. Now we need to add this bacteria, and you'll see that this is a positive control. So we're also going to want to use the positive control bacteria on this. And really, I could have used the same loop to do this because I'm going to use the exact same plate here. Positive cells. Go. Streak that across. Be nice and gentle here. Give another quick. You can do 3. You can even do 4 across. That's fine. Okay, there we go. Put this one in my inactivation bag. Let's look at the next one here. So now we've got E. coli. This one does not have ATF1. It does have the isoamyl, and it has chlor. And so you'll see this is another negative control. And we need cells that don't have the ATF1, meaning the non-positive controls or experimental, but rather the negative control cells here. Okay? So let's get another loop here ready. Take it through. Tube here. And the lid. Tip, 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 just under the surface, right? You don't need to go too crazy. And just at a nice sharp angle across the plate. Turn it 90 degrees. Do it again, and heck, let's just go once more here. Make sure we get lots of cells. Great, there's the third plate done. Put that in my inactivation bag. And then we have one more plate left here, and this is your experimental sample that's going to go on there. Again, you want E. coli with ATF1, the substrate, chlor, and then, yeah, your experimental. So that's these ones here. Let's get another loop. Open that up. Let's get a colony. These are a bit big colonies. They've been incubating for a couple of days, so no matter. Maybe smaller. We haven't incubated them as long. And let's streak that across, 90 degrees. Heck, let's do it a little bit more here. Get more cells on there. Okay, let's put that in our inactivation bag. Let's put the lid on our tube of our negative control cells, and we are good to go. So just like in other experiments, you now put the humidity chambers on top of these and put them in your incubator. And you want to incubate them for Usually 48 hours is best. That way you get lots of bacterial growth, and you also get good expression of the ATF1 enzyme. Okay, so 48 hours is pretty good, 72 hours is pretty good, and we'll check back in then. See you later. Hello, we're back. It's been about 48 hours since we streaked cells onto our Smell It Kit Petri dishes, and so today is the reveal, and I want to make just a couple of suggestions and remarks to close up the experiment. So I guess one thing you'll notice when you look at your Petri dishes is there are quite a lot of cells across the surface of the agar, which is exactly what you're hoping for, because again, the substrate, the isoamyl alcohol, which was put underneath this little paper circle here, has been constantly diffusing out of there into the agar, ultimately to the surface where those bacteria are. It's absorbed into those bacteria, and then the enzyme, depending on the Petri dish, if it has it or not, will convert that substrate into a different smelling product. Okay? So you should see pretty good coverage of cells on the surface of the plate. Okay. From a practical experimental standpoint for the reveal, one thing to note is that right now, or after you've incubated your plates, you don't want to open them all right away, because you're actually going to release an accumulation of that product, of that smell from the plate. Okay, so when they're closed, they actually do a pretty good job at holding in that final product smell. And once you open the Petri dish, you're releasing that smell, that product, and it dissipates, it goes away. And then if you put on the lid and then open it right away, you're actually mostly going to be either smelling the substrate, or there's not going to be too much smell. Okay, so it's important to note that you kind of got to prepare, be ready with all the Petri dishes so that you can smell them in series. So you can smell one right after another and see if you can distinguish differences between those plates. Okay, it will take a couple of hours, most likely, to replenish the final product smell within those Petri dishes. Okay, so keep that in mind. And the third recommendation I have is that you do this test blindfolded, or just with your eyes closed, and have a friend, a teacher, one of your bench partners open up or choose a Petri dish and open it up in front of your nose so that you can kind of guess what these different smells are. Okay, there are overlaps between the smells because you still have substrate within the positive control plate, for example. So there is overlap, but we find that it's more fun and quite surprising. Usually, in most cases, people get it right with a blindfolded test in guessing what the different Petri dishes are. Okay, so that's a recommendation. And of course, when you're smelling the Petri dishes, don't get too close. You don't need to get too close because you don't want to have your nose touch the surface of the plate and get bacteria on it. Okay, if that happens, which it shouldn't, you should just go ahead and immediately just wash with soap, but that should not happen. So avoid getting the Petri dish too close to your lab mate's nose. All right, well, I hope you learned a lot about enzymes, about experimental controls, and got your engineered cells to create a smell for you.